What happens when you get a cut? Your blood clots and the wound eventually heals. We often take this process for granted, but there's a lot of communication that goes on in your cells for this to happen. When you get a cut, cells release signaling molecules, activating the palate-derived growth factor signaling pathway, also known as PDGF. PDGF stimulates cell division, forming a blood clot to stop bleeding and promote healing. There are 5 different hydrophilic signaling molecules in PDGF. These are PDGFA, PDGFB, PDGFC, PDGFD, and PDGFAB. These signaling molecules bind to 2 tyrosine kinase receptors, or TKPs, alpha and beta. These receptors are used for cell-to-cell communication, playing an important role in regulating cell growth. TKPs are located outside of the cell membrane due to its signaling molecules being hydrophilic. In TKP signaling, PDGF ligand binds to an inactive protein receptor, activating through a process of dimerization causing phosphorylation. Phosphorylation is when a phosphate group from ATP is added. This process converts ATP to ADP. The phosphate groups attract signaling molecules in the cytoplasm, initiating the signal cascade. An advantage of using tyrosine kinase receptors is that they offer rapid cell response due to their dual structure of having both external receptors and internal protein kinase. However, they have short-lived response and can be prone to overactivation by mutations, potentially leading to conditions like cancer. PDGFs, composed of disulfide link-tied polypeptide chains, are synthesized in response to thrombin, released by palates and endothelial cells. Receptors have 5 extracellular domains and 1 transmembrane domain, with intracellular sections including juxtamembrane domain, tyrosine kinase domain, and carboxyl terminal tail. When signaling molecules bind to its receptor, it induces dimerization, which is a chemical union of 2 identical proteins, which activates the intracellular tyrosine kinase regions via autophosphorylation. After binding, the ligand-receptor complexes stabilize via interactions between them. Removing part of the carboxyl terminal tail of the beta receptor causes receptor activation. Additionally, inhibition of the kinase domain causes structural changes. Signaling begins with tyrosine phosphates inactivated via cytosine residue oxidation. Receptors bind around 10 families of SH2 signaling molecules, which activates various different signaling pathways. The GRB2/SOS pathway activates the mitogen-activated protein kinase cascade, implicated in cell growth. The PLC/Y pathway generates inositol triphosphate and diacylglycerol, mobilizing intracellular Ca2+ and activating members of the PKC family. The P13 kinase pathway mediates actin reorganization, chemotaxis, cell growth, and anti-apoptosis. PDGF receptors are expressed in wound-healing cell types like fibroblasts, smooth muscle cells, neutrophils, and macrophages. Stimulation recruits receptor-expressing cells to wounded areas with responses dependent on alpha or beta receptor types. Fibroblasts and smooth muscle cells express mainly beta, while human palates express alpha. PDGF activity induces cell growth, shape changes, mobility, and chemotaxis of various cell types. Chemotaxis is directional cell movement along concentration gradients. Macrophages produce additional growth factors, which are crucial for healing. PDGF signaling also stimulates production of matrix molecules such as fibronectin, which promotes the spreading of pallets at the site of injury and migration of neutrophils, fibroblasts, collagen, and etc. into the wound region. Overall, it speeds up the rate of healing, and the signaling persists until the pH decreases, which causes endocymes endosomes to fuse with lysosomes, ending the pathway, also known as termination. Cyclic AMP dependent protein kinase inhibits pathways via phosphorylation. Stimulation of fibroblasts or mesothelial cells leads to a decrease in the expression of the alpha receptor. The protein kinase C is also involved in this negative regulation. Tyrosine kinase receptor mutations, even a single base switch in an amino acid sequence, can cause permanent activation, leading to overactive signaling and continuous creation of diseased cells. This contributes to the development of various diseases, notably cancer. In chronic monomyelocystic leukemia, a mutation fuses PDGF beta receptors with a TEL gene, which causes overactivation. One treatment option includes in using imatinib, an exogenous antagonist that blocks abnormal kinase receptor proteins. Imatinib works by binding to mutated tyrosine kinase receptor proteins, blocking signaling molecule binding and inhibiting the pathway. Since PDGF stimulates cell proliferation, tumor growth is halted. In summary, when a cut occurs, thrombin and PDGF signaling molecules are released, binding to PDGF alpha and beta receptors, which activates the pathway. Autophosphorylation follows, which converts ATP to ADP and binding phosphate groups to receptors. Then, oxidation initiates the pathway. 10 different PDGF signaling pathways promote cell growth and repair. Lastly, when the cell's pH decreases to a certain level, the receptor releases the signaling molecules, which terminate the pathway. Thank you.